Okay, so we are now on Unit 2, which is all about the cell. And Unit 2.1 from the AP Biology course and exam description is subcellular components, which in your book, Biology in Focus, is section 4.4. And these supplemental categories, this was kind of just for me to look at some things, but you're welcome to look at it. It's going to all be pretty advanced things that have to do with different organelles and how they're related to diseases. So if you're interested in that, you might want to take a look at those. All right, so we're going to just dive right in and start talking about the ribosome. This section doesn't talk about the nucleus, but I'll talk about it in another section. All right, and I'm going to use this chart down here to talk about the various subcellular components. Ribosomes are made of protein and ribosomal RNA. And the ribosomal RNA kind of acts as like a scaffolding for the protein to kind of hang on. So if I just do an image search of ribosomal RNA, I might not even put this one in my picture, but I just wanted to kind of show yeah, so if I pick that up and kind of just bring that in, that's kind of what a ribosomal RNA looks like. We don't have to keep that in our diagram, but you can see it's RNA because it has U's in it, and it folds up upon itself based on complementary bases. So if you're looking at this sequence, this is just an interesting thing. And when we talk about evolution, this is going to be important because these sequences you're able to really look at evolutionary patterns using them. So you see here, like, these are all complementary, but then the parts that make the little circles are not complementary. And it just gives it shape, so which the ribosome could kind of be, it's almost like a hanger and a t-shirt kind of analogy where, or shirt, where if you kind of have it just rolled up in a ball, it doesn't have any shape, but if you have it on a hanger, it can give some shape to it. So I'm just going to actually go ahead and delete that because I don't need that right now in my notes. All right, and that's the structure of the RNA. And then they consist of two subunits. And remember, all organisms have ribosomes because all organisms need to make protein. So whether we're talking about a bacteria or an animal or a plant, they all have ribosomes. But there's going to be a large and a small subunit, but they're going to be different sizes between prokaryotes and eukaryotes, which is actually really fascinating to me anyways, because this is a big target for a lot of antibiotics, because if you can target the protein synthesis of bacteria without targeting the protein synthesis of your own cells, those cells will be much, you're going to have less of a chance of harming those cells. So when you take antibiotics, they're going to just kill bacteria cells. You're not going to kill the human cells. Whereas like chemotherapy is so hard on the body because those chemotherapies have to target eukaryotic cell division, whereas some antibiotics can be targeted towards the ribosome of prokaryotes. So their function of ribosomes, of course, is going to be protein synthesis. Can't type today. Also known as translation. And as far as a drawing goes, I'm just going to go, you can draw them. You can get however, if you want to be creative with this. I am not an artist, so I'm going to just look for ribosomal. And you don't have to pick the pictures I do. You could pick pictures that, especially this is going to be like review. So you know what a ribosome should look like. Or you can kind of tell from the consensus of the pictures. Just a little bit of a lesson in Cami. I can flip this around. One is showing the back and the front. I'm only interested in what the front looks like. So I'm actually going to just go ahead and crop that top one out. And then I can resize this And there is my drawing image of a ribosome. And then electron microscope, it might be hard for me to find electron micrograph of ribosomes because they're so tiny. But remember, the rough ER is studded with ribosomes. So I can just put a picture of the rough ER that's then showing the ribosomes on the surface of that. All right, I'm going to show just a quick animation of ribosomes to kind of put that in a little bit more visual context.
The ribosome is a large molecular...
So what you see here is the small subunit down here and then the large subunit of protein. We're not really seeing the ribosomal RNA, but it's kind of like on the inside like a frame. And then this is the mRNA. And now you're going to start to see these red things come in, which are tRNAs, and they're going to bring in the amino acids. So I'm just going to go, this is only a minute, so I'm going to go ahead and play it.
Factory that translates the genetic code in messenger RNA into long chains of amino acids that fold up into a protein. The gene code is read in groups of 3 letters called a codon. Amino acids are delivered to the ribosome by transfer RNA and match to the gene code in the messenger RNA.
Okay, and then this one is just kind of like a little bit more of a drawing representation, but I like this one as well.
Translation occurs in 3 steps: initiation, elongation, and termination. Translation begins with the association of an mRNA, a small ribosomal subunit, and an amino acid charged tRNA, coming together to form an initiation complex. Note that the mRNA start codon is AUG, and the first amino acid of this polypeptide and all other newly made peptides is methionine. The large subunit of the ribosome joins the complex. The ribosome contains 3 sites for tRNAs. The A, or amino acid site, accepts new tRNAs bearing amino acids. The P, or polypeptide site, is where a tRNA adds its amino acids to the growing polypeptide chain. The E, or exit site, is where the tRNA resides before exiting the ribosome. The ribosome now enters the elongation phase of translation. A tRNA with an anticodon complementary to the next codon of the mRNA enters the A site of the large subunit. The large subunit catalyzes 2 reactions. It breaks the bond between the tRNA in the P site and its amino acid, and it catalyzes the formation of a peptide bond between the amino acids from the A and P sites. After peptide linkage, the empty P site tRNA shifts to the E site on the ribosome as the ribosome translocates the length of one codon, releasing the empty tRNA. The second tRNA, now bearing a dipeptide, is in the P site. The next charged tRNA enters the open A site, and the elongation cycle repeats. Translation is terminated when a stop codon, in this case UAA, enters the A site. Stop codons do not encode amino acids or bind tRNA molecules. Rather, they bind a protein release factor, which allows hydrolysis of the bond between the polypeptide chain and the tRNA in the P site. The various components of translation then
And that's just showing you like a very small peptide, but usually there are hundreds, even thousands of amino acids long. But you can have these shorter dipeptides, tripeptides, smaller ones as well. All right, so that is the ribosome, and then we're going to move on to the rough ER. The next organelle is the rough ER, and remember that's endoplasmic reticulum, and that literally translates into little network. So it's a network of membranous tubules. In the case of the rough ER, they're a little bit flattened. And studded with ribosomes. Okay. Those are going to be just found in eukaryotes because it's a membrane-bound organelle. And the job of the rough endoplasmic reticulum is it efficiently transports newly, because it has the ribosomes on it, newly synthesized proteins to their, I don't want to say destination because that's going to be with the Golgi, proteins around the cell. So it's kind of like an analogy of having, let's say you had 1,000 cars and you had no roads and they were just all free driving around the place, whatever the place is. That would not be very efficient. It might even take longer, it'd be very crowded, it might be dangerous. So the same thing in the cell of the eukaryotic cell, it compartmentalizes functions to make things more efficient, to make things conducive to operating in the time frames that are conducive for living cells. As far as a drawing of the rough ER, I'm just going to search for rough ER. And you don't have to pick the picture I pick. You could pick whichever one you want. Especially this stuff is review, so you're going to kind of already have an idea of what it should look like. So I kind of like this picture because it shows me a couple different organelles in relationship to each other. So that's why I picked that one. And then as far as, it might be, for as far as the images go, I might use the same image when I search for rough ER. I don't find a lot that has to do with the electron microscope other than this one here that I used in the previous, but that's okay. Just kind of re-emphasize that we're looking at the rough ER there. All right, next one is going to be the smooth ER. And again, all of these that are membranous, all the rest of them are going to be eukaryotes. So I'm not going to even, well, maybe I'll just go ahead and put that in there, but just copy and paste it. Okay, smooth ER is going to be a network of membranous, I could type, tubules that are a little bit more circular, I guess if we want to describe their shape. And the smooth ER, if you remember from 9th grade bio, has a few different functions, which are all pretty interesting, I think. And so I don't know if there's necessarily any important order here, but smooth ER is going to synthesize lipids. It is going to store calcium, and it's going to be involved in detoxification of drugs, alcohols, poisons, that sort of thing. So it has all those different functions. And if you think about it, if somebody has an alcoholic, for example, or abuses drugs, this actually happens quite a bit in people who abuse pills. They have a lot of Tylenol in them, and this will cause even more problem for them than the actual opiate that, I wouldn't say no more of a problem, but as far as the cells in their liver and the detoxification, just a chronic overdose of Tylenol causes problems. So anyways, in these individuals, they're going to convert some of their rough ER, which normally has the function of producing protein, into more smooth ER. So it's going to synthesize more lipids. So they're going to get like a fatty liver. The liver is going to harden because it has calcium there, and attempt to try to promote more detoxification. And what that does is it promotes more tolerance of the medication that way. My son was on a drug when he was a very small infant, and even for his body weight, he had to keep increasing the dose because of this was happening. It's actually I think it's in the 9th grade textbook, talks about it, how it more and more of that medication is needed to have the same effect on the body because of that, that's what tolerance is. Okay, so as far as smooth ER image goes, you can pick whichever one you fancy here. I kind of like this one here because it's not, I know what it's looking at here. Number 4 is, but if you want to find one that is just showing that smooth ER, but I can tell that this is the rough ER, that's the Golgi, these are vesicles, it's a more tubular, circular part is the smooth ER. And let me see if I can find smooth ER micrograph. This Google image searches are a little bit more limited because they're the ones that you can use for Creative Commons. But I don't find them tremendously. Oh, smooth ER ribosomes. There we go. That's cool. That shows both the rough ER and the smooth ER together. I can even do something like this. I can get rid of that guy and I can do that. Maybe do it that way. Okay. All right, Golgi complex, sometimes called Golgi apparatus, sometimes Golgi body, all the same. Discovered by an Italian scientist named Golgi a long, long time ago. I think his name was Emilio Golgi or something. And it is again, membranous, a stack of membranous sacs. Okay. And function of the Golgi apparatus, you know that it's kind of our quote unquote post office of the cell. It is going to take cargo such as proteins, lipids, carbs, etc., and tag and transport to the correct cellular destination. And let's see here with our image, Golgi. See, there's Golgi, I'm assuming. I'm going to use that picture again. So they're not connected network of tubules like the endoplasmic reticulum is. Those are going to be individualized stacks of flattened sacs that are going to move things around the cell. All right, let me go back to a couple of my animations here and see what I have. Again, most of these are particular diseases. This stuff's all very, very interesting, at least to me anyways. So you might want to peruse it and see if you find anything interesting. It's going to all be pretty deep material as far as articles go. Some of this I don't think I necessarily have to watch or talk about right now.
So, what are organelles?
Oh, we know what organelles are.
Structures known as ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have.
All right, we got all that already. All right, this is the rough ER. I'm moving back a little bit, and you'll see how vesicles are going to be transported from the rough ER, butt off the rough ER, and then go to the Golgi. So this is a good place to look at this animation, I think.
In small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates.
Oh, that was kind of an abrupt end there. But that's a good look at how those organelles are related to each other. I think I like this one as well. Let me see what we have here.
Serotonin, or another neurotransmitter protein. Remember that in constitutive secretion, vesicle.
All right, that's going to be beyond our scope right now. Okay. Both the rough ER and the Golgi have been implicated in a lot of diseases like Alzheimer's disease. So one person who has Alzheimer's, there might be 100 different types of mutations that cause that, problems for that. And same thing with the Golgi. All right, let's focus on the next one, which is the mitochondrion. And we're going to talk a lot about the mitochondrion as we go through cellular respiration in eukaryotes. So I'm just going to barely kind of cover it right now. And that part should be review right now. But as far as structure of the mitochondria goes, it has an outer smooth membrane and an inner folded, convoluted membrane. Remember, those are called cristae. And an inner fluid called the matrix. That's going to be important as we go throughout the year, some of these parts of the mitochondria. And so it doesn't hurt us to kind of review it as we go. Okay, let me see if I could find a good image of a mitochondrion. I think these are usually pretty good ones. There might be some issue with it, but I'll go with that. And I'll keep it big right now while we're looking at it. Yeah, I do like this one. It has the cristae labeled, the matrix labeled, and then the ATP synthase is going to be on the membrane. And I don't think it necessarily stresses that here, but the ATP synthase, remember, are going to be the enzymes that synthesize ATP in cellular respiration for eukaryotic cells. So, I don't want us to use the powerhouse of the cell. You guys understand that, but, it's going to be involved. The mitochondrion is going to synthesize ATP in eukaryotes during cellular respiration. Let's keep it at that for these purposes right now. And then let's see if we can find a mitochondrion on the electron microscope. And a couple of things about the mitochondrion. Remember that we inherit our mitochondria from our mother, because when you look at the mitochondrial DNA, mitochondria have their own DNA. We just see the maternal lines there. So we inherit that from our mother. And a couple other things. There's quite a few diseases that are mitochondrial diseases. And most of them, not all of them, but most of them are going to be passed down from mother to child. Sometimes they're in nuclear genes that cause problems with the mitochondria. but sometimes they're also in the mitochondria themselves, and they'll be passed on from mother to child. And what's interesting is some people have very severe mitochondrial disease. It can even be fatal. And then their children, so some of their children don't have it and some do, so it's very, very unusual, sort of hard to figure out sometimes diseases, but cause a lot of very severe symptoms, because you can imagine that you need energy for all parts of your body. So if your cells don't get enough energy because there's problems with the mitochondria, either how many or the shape or how they synthesize ATP. I mean, there's thousands probably of different mutations that can happen in the mitochondria. But the diseases can be pretty severe. All right, next one that we're going to deal with here, and I'm just kind of going fast because we have so many organelles to talk about, only so much time, right? So we're going to cover more in depth on the ones that we need to at a later time. Lysosomes. So what lysosomes are structurally are just going to be vesicles. So vesicle just means a kind of generic circular bubble-like structure that is going to bubble off of something. But in the case of lysosomes, they also have inside of them digestive enzymes. So what makes something the difference between just a generic vesicle and a lysosome is it has digestive enzymes in it. And as far as their job is to break down and recycle old cell materials, whether they're parts or molecules. I'm really going to hurry up because I have a low battery. 8%. I can get this done. All right. And then one that I like to talk about with the, let me see if I can move up to it for lysosomes is Tay-Sachs disease. Let's see what I have here. Yeah, this is a good one. And it's short too, I think. Okay, so there's our lysosome, and it's supposed to have digestive enzymes in it. There's different types of lysosomes, so some in the brain might have different enzymes than the ones in the liver, etc.
In the neurons of infants, glycolipid ganglioside GM2 is anchored in the lysosomal membrane and prepared for degradation by beta-hexosaminidase A, HEX-A enzyme. Normally, GM2 activator protein is required to extract the glycolipid from the membrane to form a water-soluble GM2 activator complex. This complex is then recognized and bound by HEX-A enzyme. The alpha subunit of the enzyme hydrolyzes ganglioside GM2 in preparation for degradation. However, mutations in the HEX-A gene, which encodes the alpha subunit of the enzyme, results in the inability of the enzyme to interact and hydrolyze the glycolipid. Accumulation of ganglioside GM2 leads to swelling of the lysosomes, causing the neurons in the brain to appear distended or bloated. Eventually, loss of function and neuron cell death occur.
And if you remember about Tay-Sachs, when I showed that in 9th grade, Tay-Sachs is always going to be a fatal disorder, usually in the first 5 years of life. So it's a very terrible disease, but I talk about it now because it's a lysosomal storage disorder. So that's what LSD here stands for. So let's go down and then finish up. Let's see how many 6% I could do this. All right, so drawing wise, something like this is fine. See if we can find an electron micrograph of it. I kind of have to know what you're looking for here, because like this is microtubule, so that's not lysosome, and that was the Golgi. May or may not be able to find one. I mean, it's not the best picture, but I guess I don't have any reason to think it's not a lysosome. That looks like a nucleus, but that might be a lysosome. All right, it's kind of a boring picture because it's just circular. All right, and then vacuole. So vacuole generally is a storage vesicle. And okay, so in plants, they have a large central vacuole that's going to be involved in storing, in plants, going to be involved in storing water and ions, sugar in plants. And then in animals, they store like other things. I mean, like for example, glycogen storage vacuole in liver cells. And let me see, as far as vacuoles goes, what I can find. This is taking too long. I should already have these going. Okay, maybe I'll use something like that. And then let me see if I can find one that for electron microscope. Something like this is fine. Yeah, that's good. Nice because you can see the chloroplast there too, and the mitochondrion. Look at that. And then finally for these notes, and I knew this would be a long one, but I kind of tried to hurry up and go through it quickly, is the chloroplast. And the chloroplast structurally is going to be very, I wouldn't say necessarily similar to the mitochondria because they're actually really different from each other. But as far as the structure goes, they have an outer membrane, and then they have inner membranes that are in stacks of flattened membranes called thylakoids, which are going to be the site of photosynthesis, light harvesting. and also going to be the stroma, which is going to be is the fluid. And we'll go into much more detail of photosynthesis later, so I don't right now have to go into too much of that. But what I already did here, talking about photosynthesis, which again, remember, is going to be using harvested light energy, water, and CO2 to produce sugar. And then our kind of drawing image of our chloroplast. 1%, I'm gonna get this done. Again, you don't have to pick the same image as I do. And if this goes dead, I'm probably not going to start re-recording. So you'll be on your own for finding your image of your thing. So that will be all that we're going to talk about today if this cuts me off. I hope that you had learned a little bit review here. Look through some of the animations that I posted there too as well. This is a whole plant cell.